Okay, so here we're going to talk about viral vaccines. And so there's a list of vaccines that are commonly used or in special situations. And of course, we can add to this SARS-CoV-2. SARS-CoV-2 is quickly becoming a vaccine preventable disease. But you can see here that we have a bunch of viruses that are targeted by vaccines for protection. And many of these are going to be our traditional live viruses. You can see that we have a lot of live. And then we have some that are going to be killed viruses. Sorry. So we have some killed. And then we have some that are sort of alternative or weaker. We have subunit vaccines. I don't see our toxins here, but we have toxoid vaccines. But by and large, live is still going to be the more dominant because it is the most potent of the vaccines. Now, immunity, as we know, is going to come in two forms. We have active and we have passive immunity. Active immunity is where your immune system is going to respond either to an infection or to a vaccination and develop an immune response to it. Passive immunity is where you're given an immune response, either from mother to child in maternal antibodies or through monoclonal antibody therapies, or the transfer of convalescent serum from a patient who has recovered from the infection to a patient who is suffering from the infection, such as what we're seeing with SARS-CoV-2. Now, herd immunity is really how much of the population do you need in order to protect the susceptible. So the key for here is you're protecting the susceptible. So healthy people, from a public health standpoint, should get vaccinated because sometimes, for whatever reason, even if you vaccinate the susceptible population, they might not respond very well because of comorbidities that are occurring in their bodies. So with respect to herd immunity for measles, it is known that you need upwards to 98% of the population vaccinated. Some might say 95. It just depends on the population. And then for influenza, this is about 80% healthy, 90% high risk. And these are over the age of 65. Okay, and then SARS-CoV-2 is probably going to be somewhere closer to influenza. It's probably not as contagious as measles. Measles is the most contagious disease known to man. And so what herd immunity means basically is to prevent endemic measles. That means measles actually replicating and circulating through the population. You must have 98% of that population vaccinated. And so it's a very high number. However, you only need one vaccine for measles. It's a very stable virus. Now, by saying stable, that does not mean there aren't variants out there, but it's just that there's one serotype. Serotype means our antibodies recognize all the variants. Currently with COVID, there is also only one serotype. Even though there's mutations, our antibodies still recognize all of those mutated viruses. So with respect to herd immunity, you're really going after serotypes. Now with influenza, why do we need annual vaccines? Well, there's two main reasons. One is that there are animal reservoirs. And by having an animal reservoir, it makes it very difficult for us to eliminate it. We were able to eliminate smallpox because smallpox only infected humans. There were no smallpox viruses hanging out somewhere in the animal kingdom. But with respect to flu viruses, there are flu viruses that affect swine and avian, which are the most important for us. The second is something called the original antigenic sin, OAS. OAS. And so for influenza, you get drift, and this is subtle changes. And you get a shift, which are leaps, big change. And so what our annual vaccine protects against is drift. Now, at sometimes it can also protect from those shifts, but really it's there to protect against drifts. So let's look at our drifting virus. So let me go to black. So we have year 1. Okay, we'll go up 5 years. And so we have our influenza circulating, this little pie chart. I should have moved that guy. Zoom here. All right, so there's a little pie chart. And now we're going to have different colors. So for year 1, we're going to have a blue quadrant. We're going to have a red quadrant. We will have an orange and a green. Okay, each of those colors represents an antigen. And so the first year you get your flu vaccine, you're going to have anti-blue. You are going to have anti-red, anti-orange, and you're going to have anti-green. Okay, and so year 2 comes along. And in year 2, you have your red, you have your blue, and you have your orange, and now you have a pink. You lost your green. And so what happens? Let me do this. You now have a booster response, so these are all boosted, but you have no pink. And the next year, now you have pink, and you have your blue, and we have a brown, and we have, let's put in here, purple. And so in this case, your blue responds and protects you. But you have no pink, you have no brown, and you have no purple. Now in the 4th year, you can imagine what's going to happen. Actually, it'd be easier if I just copy and paste this guy. Oops. Sorry. Right, so in the 4th year, we are going to get rid of this blue guy. And now this time you have again your pink, your brown, fluorescent green. And now you lost all the original ones. The blue, the red, the orange, and the green are no longer by year 4 part of this virus that's circulating. So now you're going to have to make anti-green, anti-purple. You're back at square one. Anti-brown and anti-pink. And so here, when you're infected, you had disease, no disease, no disease, disease. You lost all that protection because you know the variants circulating no longer have the original 4 colors. They now have 4 new colors. And the original antigenic sin, what's important about it is that your response to these first 4 colors prevents a response to these new emerging colors. And so, in theory, here you would be vaccinated to pink. And that would give you an immune response to pink. Here, you can be vaccinated to brown and purple. And that will give you that immune response. And here, you can be vaccinated to green. So again, vaccinations are going to sort of fill in the holes of how the virus is mutating so that you keep on top of these drifting viruses so you never get to year 4. So even at year 4, if you didn't have an anti-green response because you got vaccinated in between years 1, 2, and 3, you would have an anti-purple, anti-brown, and anti-pink response because the vaccine filled those in. And then now in year 5, you'll kind of reset. I'm not going to draw it out, but this is kind of a reset. And you have your immune response depending on the variant. Is it protective? Is it not protective? Does the vaccine have to fill in another gap, etc.? Now, conjugate vaccine is where your immune system is kind of tricked into responding to an antigen it normally would not respond to. So this was shown with Haemophilus influenza. And it's also used for other capsule type vaccines. So this is a bacteria. And you have a capsule. Now a capsule has no proteins. And so we know, okay, this is what we know, that you have a capsule antigen and you're going to stimulate IgM response. And again, this has no protein. And so you get no CD4 response. So this leads to no help. And the only response you can have is IgM. IgM does not form memory. IgM cannot neutralize. It cannot cross the placenta. Only thing IgM can do is activate complement. And so having just an IgM response is not very protective. So we also know if you have an antigen that's a protein, you get IgM plus CD4, and you get help, which gives you IgG, for example. Okay, we know this. These are 2 things that we know. So the people who were trying to develop a vaccine for Haemophilus influenza were like, well, how do we take what we know and apply it to this pathogen? And so what they did is they took the capsule, which has no protein, and they linked it to tetanus toxin. which is a protein. And so this is a link. And so what you get is you get anti-capsule IgM, and you get anti-tetanus CD4, and this guy can help the anti-capsule, and you can get IgG. And you might say, how can they help? Well, it's because since this is linked together, your B cell on your MHC class 2 will express the tetanus peptide, even though it's releasing IgM that's for capsid. So you're kind of tricking the immune response. You're saying, I want this capsid B cell to present tetanus peptides to a CD4 tetanus-specific T cell. And then that CD4-specific tetanus T cell thinks that it needs to help this B cell, and it does. It helps the capsid-specific B cell switch from IgM to IgG. And that's how the conjugate vaccine works. So and then finally, vaccines. Who's best, who's the worst, or does it matter? Well, you have live attenuated, and this typically means can replicate. It's usually about 1 round. And then you have your killed, and this is no replication. So here's 1, here's 2. And then you have your proteins, such as your toxins, and this is no replication. You have your nucleic acids, your RNA, and this is no replication. And then you have your subunit. Well, I'll put conjugate first. Your conjugate and your subunits. And this is typically protein, and this is also protein-based with plus a sugar or fat, for example. And so if you look at all of these, and the 2 things you're mostly interested in are efficacy and safety. So this has high efficacy, and it has lowest safety. The killed has higher, oops, did I do that? The killed will have relatively high efficacy, and it'll have better safety. Your protein toxins have high efficacy and are very, very safe. They don't ever cause disease. Nucleic acids, we'll see where this goes. I'll put it here, not sure. And safety, they are relatively safe. They have side effects, but they're relatively safe. So I'll give it 4. Conjugate vaccines, efficacy, they are very good at capsids, and they are very safe. Subunit vaccines are very weak, but they are the most safe. So you never have diseases associated with protein vaccines.